So hi everybody, I'm Kelsey. I'm a postdoc at the University of Minnesota, and I'd like to thank the organizers for giving me the chance to speak to you all today about genetics and genomics of human milk. Lactation is the defining trait of all mammals. Successful lactation has been essential for the survival of our species. And yet we know very little about how human genetic variation impacts lactation or human milk composition, or how variation in milk composition impacts infant traits, such as the infant gut microbiome. So maternal factors like genetics, diet, and health all play a role in shaping lactation and milk composition, but we don't know the relative importance of these factors and their impacts on specific milk components. In particular, there have been very few studies of the genetic impact on human milk. And for the vast majority of milk components, we don't really know what the role of maternal genetics is. Similarly, we don't know very much about how variation in human milk composition impacts infant health and development, for example, the infant gut microbiome. Differences in milk composition have been associated with a lot of different infant traits, including, of course, the gut microbiome. But in general, these associations have come from studies where a few or single components are tested against a single infant outcome. And for most milk components, we don't have any idea how natural occurring variation may impact infants. So I always like to highlight the complexity of human milk composition, which a lot of people don't think about if this isn't something they study. So in addition to the macronutrients of fat, mostly in the form of triglycerides, sugars, mostly lactose and proteins, milk contains hundreds of what are usually called bioactive factors, such as antimicrobial peptides, antibodies, and maternal cells. And these bioactive factors are thought to have diverse functions in the infant, such as guiding immune and intestinal development. Milk also contains prebiotic sugars called human milk oligosaccharides, which make up about 15% of the dry mass of milk and are not digested by the infant, but they provide food for microbes in the infant gut. And HMOs are synthesized de novo in the lactating mammary gland from lactose. And of course, milk also contains its own microbiome. So today I'm going to tell you about a project I've been working on where we collaborated with the Milk Study, which is an ongoing multi-site study at the University of Minnesota and Oklahoma. And I used the data that they collected for about 200 breastfeeding women who were recruited during healthy, low-risk pregnancies. And they have maternal and infant clinical data, demographic information, and also some milk composition information. So within this resource, we used 1 month milk samples to do RNA sequencing to profile gene expression in milk, whole genome sequencing to profile maternal genetic variation, and also the human milk oligosaccharides in a subset of these moms. And then at 1 and 6 months postpartum, the Milk Study also had generated shotgun metagenomic sequencing data from infant fecal samples. So today I'll tell you about my work to integrate these genomic data sets to first perform an EUTL analysis of human milk, which means I looked for genetic variants that are associated with gene expression in milk, and then to look at those genetic variants and see if they also associate with the composition of HMOs in milk, which again are the sugars that feed microbes in the infant gut. And finally, to look for relationships between milk gene expression and the infant gut microbiome. So just to put the gene expression data in context, this is a PCA showing a few of our milk samples in black compared to many different tissue sites from the GTEx consortium. And you can see that the milk clusters in the middle near other secretory types of tissues like kidney, colon, and pancreas. And this is just showing that milk is profiling the biology of the lactating mammary gland. So we identified milk EUTLs at about 2,700 genes. Again, this is associations between maternal genetics and gene expression in milk. And this is about what you'd expect based on the sample size that we have. So we compared these genetic variants that influence gene expression in milk to those that had been seen in other human tissues, again from the GTEx resource, and found that the tissues that were most similar to milk gene expression were minor salivary gland, stomach, colon, so other secretory type tissues, even more than resting breast mammary tissue. So this is highlighting that genetic regulation of gene expression in the lactating mammary gland is quite distinct from that of the resting breast tissue. So next we wanted to see if these genetic variants that influence milk gene expression are also influencing milk composition, because this will give us information about the genes that are playing a role in shaping milk composition. And specifically, we were going to look at the composition of the human milk oligosaccharides, or HMOs, in milk. This plot is showing the composition of HMOs for about 50 women from our study. And you can see that they're broken down into two groups, non-secretors and secretors. So each bar here is an individual person's milk sample, and the y-axis is showing the concentration of 19 different HMOs. And so the reason they're broken down into these two groups, non-secretor and secretor, is because this is something that was previously known, that if you are a secretor, which is determined by if you have a functional FLT2 enzyme, then you can create some HMOs, such as 2FL, which is shown here in navy blue, that is basically absent from non-secretors. So there's one previously known very strong genetic variant that determines the composition of HMOs in your milk. But you can see that even within these secretors, for example, there's a lot of variation in terms of the total amount of HMOs you have and also the different levels of each individual HMO. So HMOs have been shown to play a role in promoting a healthy gut microbiome. So hopefully understanding how maternal genetics impacts HMOs will help us understand how milk variation can play a role in shaping the infant gut microbiome. So we look at the maternal genotypes at our milk EUTLs. Again, this is genetic variants that modify milk gene expression. So I'm going to show here the FLT2 gene, which I mentioned is what determines secretor status and has a strong influence on the HMO composition. And this is showing that individuals with the G allele have much lower expression of this enzyme. And we see, as expected, that in their HMO data, the individuals that are non-secretors have basically no 2FL, this particular HMO, in their milk. But we also discovered some new relationships. So for example, at this gene GCNT3, we identified a maternal genetic variant that each T allele increased the expression of this particular gene in milk. And we saw that it was also associated with the concentration of this HMO FLNH. This makes sense biochemically, given what we know about the GCNT3 enzyme and this HMO, but the association of this genetic variant with GCNT3 expression and this particular HMO is a new finding. So finally, we hypothesized that milk gene expression, which we're using as a proxy for the activity of the lactating mammary gland, can drive differences in milk composition, which might impact the infant gut microbiome. So because of the high dimensionality and correlation structure in these two data sets, we used an approach called sparse canonical correlation analysis to integrate these data sets, which results in identifying correlated sets of genes and microbes across these two data types. So one main result from this analysis was we found that expression of the JAK-STAT pathway in milk was negatively correlated with the relative abundance of Bifidobacterium infantis, which is a beneficial microbe in the infant gut that's associated with a wide variety of positive health outcomes. So this plot is just showing like a network of the relationships across these genes, shown in green in the JAK-STAT pathway, and then in blue, the B infantis relative abundance or growth rate. And the take-home message here is that the lines connecting B infantis to the JAK-STAT pathway genes are all dotted lines showing that they're negatively correlated. And this pathway is interesting because it's involved. It's important for normal mammary gland development and epithelial differentiation towards lactation, but it's also can be activated as an immune response to infection and to mastitis, for example, which is the common infection of the mammary gland. And we also thought that these genes were correlated with the cytokine IL-6 concentrations in our milk samples, suggesting that there could be a signature of inflammation here that might be potentially driving these differences. But we don't know for sure if these moms had a mammary gland infection or what might be driving this possible immune response. So to summarize today, I showed you that we can use maternal genotypes and milk gene expression to identify genetic influences on gene expression in milk, and that these EUTLs are useful to identify the genetic variants in genes underlying differences in milk composition, such as these prebiotic sugars in milk. And I also looked at patterns between gene expression and the infant gut microbiome. Moving forward, as additional and larger data sets become available, it will be great to be able to follow this path all the way through from maternal genetics to the infant gut microbiome or other infant outcomes, and to understand how maternal genetics and the mechanisms by which milk variation shapes infant health and development. So with that, I'd like to thank my mentors, Frank, Ron, Ellen, and Cheryl, our collaborators, all the milk study participants, and our funding sources. And if you're interested to learn more about this, we have a preprint available at this QR code. So thank you very much.